All right. We got the band together, and this is the last session. However, please stay for the presentation awards. Otherwise, Dr. Rahal will be very upset with me if I didn't say that to you now. But often I'm put in this position of running a panel where technologies, like you just heard, are breakthrough or futuristic, and often, they're 10% proven at best. But that's where the exciting part is. And I'm joined by some esteemed colleagues of mine, many of whom I've known for many decades. So let's start over there in the far corner is J.P. Hubschman. He is a professor of ophthalmology and engineering at the Stein Eye Institute at UCLA. J.P., welcome.
Thanks.
Okay. Next is Pierre Bayardin, from the CEO of DORC. Welcome. DORC's been a cornerstone for ophthalmic surgery for a very long time, so welcome. And my good friend Paul Hallen,
Mark.
for VP and Global Head of Retina at Alcon. Many of you know him. And of course, my colleague at USC, Aaron Nagel, who is assistant professor. So let's get started. The way I was going to do this is ask some questions about previous talks and also ask you something about what you're thinking. So one of the things, so a little bit surgical, since you guys are interested in that. So we've heard about gene therapy, cell-based therapies, but let's start with gene therapy. And it requires delivering a small volume of fluid to the back of the eye. So Pierre, why don't we start with you? We're very used to delivering large volumes of fluid when we do eye surgery or cataract surgery, but this is a different volume. It needs to be more precise. We have to prevent reflux. Thoughts on how you are thinking about it? I know DORC is working on this. I haven't seen your new system, but maybe you can talk a little bit about that.
Yeah, sure. Thanks, Mark, and thanks for having me. Yeah, we're very proud because we're introducing at ASRS today our first FDA-approved subretinal microinjection system. So it's a system that is that comes with foot pedal integration, where we've got very high level of control of pressure, plus minus 1 psi, for sub 0.1 milliliter injections volumes. So that brings the surgeon autonomy. So you don't need to have someone holding for you the syringe for a very long time. So it brings stability, safety, control of the injection, control of the volume as well. So
right.
That's what we're bringing today. We're very, very proud of that.
Thank you. Paul, anything Alcon's doing on this front?
Yeah, we've been able to do this with 41 gauge cannulas going all the way back to macular translocation. So the market is very small. The technologies out there, we're looking at technologies out there that are very, very good. Not only deliver it very precisely, but also can give you signals on what you do. So the market's so small, though, the investment probably is wiser in other areas.
Yeah, well, market is small right now, but it could obviously grow. So, Aaron, can you've done obviously with Luxturna and others, you've done subretinal gene delivery. What are your thoughts from a surgical standpoint? How do we need to do it? And some of the subretinal changes, so Aaron has just published some really good work where after injecting in the subretinal space, you're seeing this RPE atrophy, and looks in areas like geographic atrophy, the paper just coming out. How do you think we can inject it? And then second question, is this related to the injection or something else that's happening, the atrophy that is?
Yeah, great question. So getting back to the first part of the question with regard to the surgery, I think having full foot pedal control has been a great advance, and many of the surgeons in the US are using that. And I think another welcome advance might be to have volume tracking so that the surgeon can have an audible cue as to how much they've injected. Right now, most sites, I believe, are just using another person with a cell phone light, trying to figure out what number we're at in terms of the injection, and these are sub milliliter volumes. So that's tricky. So we welcome innovation there. when I originally saw this phenomenon of basically geographic atrophy and a perifoveal distribution in these patients, I thought initially, since it hadn't been described in the phase 1 and 3 trials, that this could be something surgical that I was doing. And obviously other sites are seeing it, it's not surgeon specific. But I think that's why it's so important to have, really nice control over the delivery, so that's not a factor. What we're seeing now, it's not so much the surgical delivery, but we're finding that the patients who get it are in that 5 to 15-year-old range, and it seems like it may be a dose-limiting toxicity where these RP cells are essentially getting burned out by production from the RP65. These are the patients that have the highest and best improvement in FSC, and then those are the patients that end up burning out those cells. So it may actually be related to the vector.
So kind of overdriving those resident RP cells. JP, how do you like any thoughts on microdose delivery to the back of the eye?
So I think that I would go back a little bit, and I think that there are many surgical tasks, in particular during retinal surgery, where the human body presents some limitation, right? The sensing, the motor skills, and even the processing, the ability to process multiple images at the same time, like digital microscope, OCT, is difficult for a human body and human surgeon. And so I think that those tasks can be done by a human. However, I think that robotic technology with a fully integrated platform, not just like a precise robotic arm or a better visualization system. I mean, each part brings something, but what I envision is really having a fully integrated platform where you have a enhanced visualization, you have enhanced manipulation and some processing capability to assist the surgeon during those surgical tasks, or even replace the surgeon at some point. We can automate some of those surgical tasks. Right. The subretinal injection can be easily automated, meaning the system can use artificial intelligence to differentiate all and the anatomical structure, assess the progression of the subretinal injection, and just do it with human supervision. Right. But I don't think that today it makes sense anymore that the human surgeon should do it. I mean, it's like the technology is there, and those tasks should be assisted with enhanced visualization, enhanced precision, and the whole platform.
JP, you're a global thinker, and you took away my other two categories, but not quite fully. So thank you for doing that. But the other two categories I wanted to talk about is visualization and motor skills, and which you're pointing to. I mean, we did a study a long time ago that showed that 80% of what we do in the eye, we can't as humans feel. We think we're feeling peeling the membrane. We think we're feeling this or that, but we just can't. And so we're really only reliant on visual information when we operate, whereas other surgeons, general surgeons, orthopedic surgeons, have a tactile feel, but we don't. Cataract surgery, maybe a little bit more, but in retinal surgery, not so much. So robotic surgery is very important. I believe it's an interesting area, but industry has talked a while about it, and there's been a little bit of a hesitation. There are different types of robots. We worked 20 years ago with an assistive robot where basically you took a robot and then we called it a steady hand, where basically you went in and through haptic feedback, it steadied your hand when you were at the target, versus like what you're describing perhaps, or in part you were describing, was what's called a primary secondary robot, where it's more like the Da Vinci. You're sitting in a remote place and the robot is doing that. And I have a third version of it, which we're working on, which is very interesting, which is nanoscale, origami-based. Origami, we think of is kind of foldable, Japanese art, but we're working on some technology with Caltech at a nanoscale origami robot, which is doesn't need a lot of powering, because power is a big issue here. But robotics, let's just ask Paul Hallen. He's seen this assistive robot technology. Where is robotics? Steve Charles talked about it 2 decades ago. Where are we?
Well, I think you got to look at the big picture of retina surgery. So over the last 30 years that I've been involved, it's really been a focus on improving the tools because they need a lot of help. And of course, we're cutting 5,000% faster than back then. The forceps are now disposable. We got MIVS entry. So we've done a great job on the tools, but now you say, well, what's the weakest link right now? And that's that precision. I think precision is not quite enough. Robots bring that, but it's got to bring efficiency, lowering adverse events. I agree that some steps you can automate. You may be able to automate the entire cataract procedure, but I think only steps in the retina. And then beyond that, I think with the 2 sclerotomies, we're really talking about 2 robots, right? And so 2 robotic arms. So I think now's the time to look at it very seriously. We're doing that. And I think it's potential to have a big impact. Also, I think that, reattaching otherwise healthy retina is one thing, but then perhaps maybe repairing diseased retina, it could play a role.
Right. Pierre, Dork, robots, comment?
Yeah, we'll look at the space at different technologies. So, as much as we're proud of Inicio, our new subretinal injection system, we believe it's definitely not the end of it. It's certainly an area for subretinal injections where, this is, to me, the primary injection, the primary indication today. Focus. Anything that has to do with image-guided surgery, has to do with partially with robotics. My personal vision is that I'm much more into the surgeon-assisted kind of robots, as those big elephants and that do things for the surgeon. So there might be some evolution there, I believe, with respect to the current systems that are about to enter the market.
Yeah, JP?
Yeah, one small comment. I think there are different types of robot that can be just the end-all, it can be co-manipulation, as you mentioned, it could be teleoperation, it could be automation. I think that whatever you choose, it needs to be bringing, obviously, better outcome for the patient, but it should also be well integrated in the surgical suite, meaning that if you have one robot which is dedicated for just a small task, and it takes time to set it up, and it won't be accepted and adopted. So it needs to be kind of multitask, and it should be well integrated, so it's actually improving the throughput. Yeah.
It's very interesting, yeah. We're working on a very complicated issue, which is basically an injectable nanoscale robot that then functions and does part of the surgery. We're working with Harvard and Caltech on that, but it's a challenging problem because you don't have the conventional robotics, but this is an injectable nanoscale robotic. But we don't have enough time to go over that technology in this panel. But let's talk about visualization, because I think that is interesting. That's another sense, and the sense that we really rely on here. So Ingenuity, we heard about bionic, the heads-up display. So Paula, I'll start with you. I mean, beyond ergonomics, which now, we're getting faster retinal surgery than, when I'm sitting there, I'm already having that arm pain. Faraz was just telling me it doesn't relieve you of putting your hands and being in that uncomfortable situation. Ergonomics, teaching, I get it. Where is this, and where do you think Ingenuity and other such systems could take us?
So I think we need to have a mindset change because digital eats analog for breakfast, right? Your iPhones, yada yada. So, how do we get there? And I think that first of all, the image quality already is amazing. Is it better in every aspect than the microscope? No. We need to solve those problems. But depth of field, magnification, the ability to use low light in retina surgery is great. Axial resolution, so it's pretty incredible, but we need to improve upon that. And then there's 2 platforms, and we're going to really benefit from the electronic industry, Silicon Valley, basically. And it kind of bifurcates into flat screen displays, and there's a whole lot of technology going on there. The 4K OLEDs are amazing, 8K. On the other side, we got wearables, and I applaud Bionics and Occutrex and people that are trying to push forward wearable headsets. But right now, they're a detriment. They're heavy. I think when you have an artificial horizon, when you're totally enclosed, like in the bionic system, that needs to change. I think, Occutrex is looking at something that is basically a hologram, a bright view in front of you, and I can look over and see Mark. So that's probably right, but we need to get to that very lightweight, 200 grams or less, so we're not having ergonomic problems. And I think you're going to want to have both choices, because both technologies are parallel advancing very rapidly. So the future is really bright for digital, and analog is dead man walking. So if you're using an analog microscope by the end of the decade, they will call you a dinosaur. So you might as well change now.
Okay, that's a pretty strong statement. So, Aaron, are you convinced to throw away your microscope and go all digital?
I mean, to be honest, the microscopes we use now are amazing, a beautiful visualization, but for procedures like gene therapy, it is nice to have the OCT there, but I find it to be not that useful because, my eyes can only look at one thing at a time, so it's a little bit hard to be looking at the scans of the OCT while I'm looking at the retina and so on. So I think it would be a welcome advance to have kind of more kind of native integration of OCT in some way to augment our view so that we can see what's going on. And
more OCT integration with microscope. JP?
So wait, quickly on the visualization, I think it's the same problem, I would say. The human body has limited sensing, and the ingenuity is bringing an amazing image. However, I think that we need to push a little bit further. We need to have the real-time OCT with real-time processing information, kind of fusing all these image modality to provide much more information to the surgeon. So it's already kind of enhancing the depth perception, and so, but there is some other steps to achieve, I would say.
Yeah. And I would add one more. I think, we're always looking at the resolution. We get aliasing when we go past a certain frequency, right? I mean, you just can't see beyond a certain wavelength of light. But we've just completely forgotten spectroscopy. And I think spectroscopy allows you to go beyond the wavelength of light you're using, so you can go beyond that and take a look. So that's the thing to add. I do, we are all, so back to Aaron, I think a question now on quantum dots. I mean, QDs are a totally different type of animal. Have you given this thought? I mean, and I'll go around the table. You guys must have thought about it. If Aaron, you haven't, that's okay.
I think it's very welcome. Great potential. I'm curious to see how it plays out. It's always a question of what's that sweet spot in terms of the safety of the quantum dots, which seem to be safe. The target population, how much degeneration the patients will have, and the mechanism of action to know kind of where it's going to fit. But I'm looking forward to seeing kind of more data, and hopefully it'll come through.
Okay, just a quick check on quantum dots, Paul. Quantum dots?
Yeah, show me the data, safety, efficacy, an open mind, and I hope it works.
Pierre, anything on quantum dots?
No, not really.
That's
no.
okay. JP?
I'm sorry, I don't.
Yeah, I think one of the things that's interesting on the quantum dots is, electrical current to restore sight we understand because you're bypassing a damage and you're stimulating the other, but what is a quantum dot exactly doing electrically to preserve those neurons? What's the mechanism? And I think that, we don't understand. It's sort of an interesting area, and we'll learn more about it. So one other technology which is very interesting is, of course, optogenetics. So again, I'll start with, this is, you've heard a number of talks, providing an additional function to a neuron which normally natively doesn't perform that function. So you're making, for example, a bipolar cell light sensitive. So Aaron, what do you think about this sort of gene therapy?
Yeah, I think it has great potential because it's kind of latching onto the existing circuitry in the retina, which still exists. Doesn't require us to create, RPE photoreceptor, layers that are kind of transplanted. It's kind of a simpler engineering problem because we're already doing gene therapy with Luxturna and others. And with targeting bipolar cells, potentially you're going to get a kind of better, more physiologic, visual perception than you might with just RGCs, for example. So I think it's really exciting, and I think it'll be welcome in our field.
Yeah. Paul, any comments?
Yeah, same thing. We meet with all these companies, and we're very interested, but I've got to see the data. And there's others, biophotomodulation, neurostim related devices. So, I believe that there is going to be probably an office-based market for some type of device-related technology. But again, we just got to wait until the data's there. They're amazing, but they actually have to work, so.
Pierre, any thoughts on these optogenetics?
I think, all that is really absolutely amazing, the progress that were made in the past 10 years. For us, we're really focused how we can help those companies to bring their product at the right location with the highest precision and safety.
JP?
So, it looks like a very promising technology, but I don't have too much experience, so I'm looking at it from outside, and I don't know much about it. So.
Yeah, I think one thing is with these, whether it's a quantum dot or whether it is optogenetic therapy, just remember the quantum capture has to be efficient. Now, why are rods and cones? They're stacks of photopigments, and they're large antennas to capture light. Quantum dot, and some of these other therapies are not giving you an antenna. So you got to look at the quantum efficiency of this, and I've done some calculations. It's very interesting to think about that, and that subsequently increases the light budget. Now, some of the new options are more light sensitive, so you don't have to blast so much light into the eye. But the other thing, from an engineering standpoint, you have to look at the quantum capture. So I think last question that I had with the last few minutes is just AI, artificial intelligence. We've heard a lot of things about that. So Paul, what's Alcon doing AI?
Yeah, so for me, why AI? I think that we need, early disease detection. So, taking a diagnostic into an ophthalmologist or OD, but having the intelligence of a retina specialist embedded in the machine is what we need to bring those patients forward early. So, for example, with Apellis and these other drugs coming out, Iveric Bio, et cetera, if we can catch geographic atrophy that much sooner, or if there are, devices out there for intermediate dry AMD, we want to bring those people in. So that's where I think it happens. Ironically, everybody can do AI, basically. So it's going to be the technology. It's going to go back to the hardware to see who wins. So, stay tuned.
Yeah. Aaron, any thoughts on AI?
Yeah, it's not really my field, but I'd say as a retina specialist, it's very much welcome, not just for machine learning and telling us what that diagnosis is, because I don't think patients or doctors want a robot or a program to tell them what's going on. But in terms of kind of organizing our thoughts, organizing our clinic flow, organizing clinical trials, we have a lot of data. What testing does the patient need? So I think that could really augment our clinical practice, because we just have so many different imaging modalities, and things can get very complex very quickly. So having some AI help with that would really be useful.
Pierre, and quickly JP.
Yeah, I think why AI? I think prediction would be great. Anytime we talk with surgeons, how would it be great if you guys would know in advance, oh, guys, in the minute to come, you're going to get a massive hemorrhage, right? So, and we've got plenty of data in our machines that we're not using properly. Plenty of data within the anesthetist material as well that we could be using. I think that's the direction.
JP?
Yeah, so AI can be obviously applicable for many, many different things, but in robotic in particular, there are two major applications, I would say. Number one is tissue recognition. So we can use AI to train models to recognize all the different anatomical structure during the surgery. So during retinal surgery or cataract, those models are working pretty well and can visualize and detect and differentiate tissue properly. So this is number one. Number two is continuous improvement. And that makes a huge difference because, for instance, if you have a robotic platform doing the surgery, everyone can benefit from the learning from every other surgery, from every other platform, meaning there's a continuous improvement on the technique. And one interesting thing is for surgical, for instance, prostatectomy today, there's more than 3,000 different surgical techniques. Right. So every surgeon, and Retina, I'm not sure, the study was not done, but probably it's the same number. Maybe thousands of different ways to do retinal surgery. And that is not really a good thing. And we can, with AI, standardize surgery and find the best way to do it. Right. And I think that this is important.
Yeah, I think I'll leave you with this one thought. CNNs, convolutional neural networks, depend really on reliable and large data sets. So here where we're talking about in our field, and especially surgical field, is sporadic data sets. So when you start to predict off a sporadic data set, things get super complicated. So I think that that's another challenge for us. But with that, our time's up. Hopefully you've enjoyed the session, and now for us for the awards.